Introduction. Medical nanotechnology is an emerging and exciting field that has the potential to revolutionize the treatment and prevention of viral infections. The use of nanoparticles as therapeutic agents offers unique advantages, such as targeted drug delivery, enhanced antiviral activity, and the ability to overcome viral resistance. In this comprehensive script, we will delve into 12 key topics related to medical nanotechnology and its application in antiviral therapies. Each topic will provide a detailed exploration of the mechanisms, strategies, challenges, and future prospects associated with nanoparticle-based antiviral delivery systems. Nanoparticles have demonstrated the ability to inhibit viral replication through various mechanisms. One mechanism involves the direct interaction of nanoparticles with viral particles, interfering with their attachment to host cells. Additionally, nanoparticles can trigger innate immune responses, leading to the production of antiviral cytokines that inhibit viral replication. Furthermore, nanoparticles can act as carriers for antiviral drugs, delivering them directly to the site of infection and preventing viral replication. The understanding of these mechanisms is vital for the rational design and optimization of nanoparticle-based antiviral therapies. Further research is needed to explore the specific interactions between nanoparticles and viral components, as well as their impact on viral entry, replication, and assembly. Nanoparticles provide a promising platform for the delivery of antiviral drugs, addressing challenges such as poor solubility, rapid clearance, and nonspecific distribution. By encapsulating antiviral drugs within nanoparticles, their stability, bioavailability, and targeted delivery can be greatly improved. Moreover, nanoparticles can protect the drugs from degradation and enhance their residence time at the site of infection. Various types of nanoparticles, including liposomes, polymeric nanoparticles, and inorganic nanoparticles, have been explored for their ability to enhance antiviral drug delivery. These nanoparticles can be functionalized to specifically target infected cells or viral components, improving drug accumulation and reducing off-target effects. Efforts are ongoing to optimize drug loading efficiency, control release kinetics, and minimize potential toxicity associated with nanoparticle-based drug delivery systems. The development of personalized nanoparticle-based therapies tailored to individual patients' viral profiles is an area of active research, promising significant advancements in the field of antiviral treatments. The surface properties of nanoparticles play a crucial role in determining their interaction with viral particles and cells. Surface modification strategies, such as the attachment of targeting ligands or antiviral peptides, can enhance the antiviral activity of nanoparticles. These modifications enable specific recognition and binding to viral proteins or receptors, leading to the inhibition of viral entry or replication. Furthermore, surface modification can promote nanoparticle stability, prevent premature drug release, and improve cellular uptake. By tailoring the surface properties of nanoparticles, their therapeutic efficacy can be greatly enhanced, while reducing off-target effects and toxicity. A range of techniques, including chemical conjugation, physical adsorption, and bioengineering approaches, are employed for nanoparticle surface modification. Advancements in nanomaterial synthesis and characterization techniques have enabled precise control over surface modifications, facilitating the development of highly efficient and selective antiviral nanoparticle therapeutics. Theranostic nanoparticles hold immense promise in the field of antiviral therapy, as they enable simultaneous diagnosis and treatment of viral infections. These nanoparticles possess both therapeutic and diagnostic components, allowing for real-time monitoring of infection progress and targeted drug delivery. By incorporating imaging agents or contrast agents into nanoparticles, clinicians can visualize the location, spread, and severity of viral infection. This information enables the precise and timely administration of antiviral drugs, enhancing treatment efficacy and minimizing side effects. Additionally, theranostic nanoparticles can be designed to respond to specific stimuli associated with viral infections, such as changes in pH, temperature, or enzyme activity. This responsive behavior ensures controlled drug release at the site of infection, further improving therapeutic outcomes. The development of theranostic nanoparticles represents a promising direction in antiviral therapy, combining diagnostics and therapeutics into a single nanoplatform. The emergence of drug-resistant viral strains poses a significant challenge in antiviral therapy. However, nanoparticles offer unique advantages in overcoming viral resistance by providing alternative routes of antiviral activity. One strategy involves the encapsulation of multiple antiviral agents within nanoparticles, targeting different stages of viral replication. This approach prevents the emergence of resistant viral strains by simultaneously targeting multiple viral components. Additionally, nanoparticles can be designed to inhibit viral proteins involved in resistance development, restoring the efficacy of existing antiviral drugs. Moreover, nanoparticles can enhance the immunomodulatory response against viral infections, stimulating the host's immune system to mount a robust antiviral defense. This approach provides an alternative avenue for antiviral therapy, focusing on enhancing the natural immune response to combat viral infections. Understanding the mechanisms of viral resistance and designing nanoparticles to overcome it is critical for the development of effective antiviral therapies. Continued research in this area will pave the way for innovative approaches to tackle drug-resistant viral infections. Nanoparticles have demonstrated immunomodulatory effects that can be harnessed for the management of viral infections. By modulating the immune response, nanoparticles can enhance antiviral defense mechanisms and reduce viral pathogenicity. Nanoparticles can stimulate both innate and adaptive immune responses, promoting the production of antiviral cytokines, attracting immune cells to the site of infection, and enhancing antigen presentation. This modulation of the immune system can lead to a more effective clearance of viral particles and infected cells. Furthermore, nanoparticles can be engineered to selectively target specific immune cells, such as dendritic cells or T cells, for precise immunomodulation. By leveraging the immune response with nanoparticle-based therapies, both antiviral activity and host immune defense can be synergistically enhanced. Research on the immunomodulatory effects of nanoparticles is still in its early stages, with promising results from preclinical studies. Further investigations are required to assess the long-term safety, efficacy, and immunological consequences of nanoparticle-based immunomodulatory therapies. In order to effectively target viral infections, scientists have developed strategies to enable nanoparticles to bypass barriers and deliver medication to infected cells. One approach involves using cell-penetrating peptides or ligands, which encourage the uptake of nanoparticles by infected cells. Another strategy involves designing nanoparticles that can cross cell membranes through mechanisms like membrane fusion or disruption. Stimuli-responsive nanoparticles can also be tailored to release medication in response to specific triggers within infected cells. Further research and understanding of cellular mechanisms are essential to refine these strategies, enhancing the effectiveness of antiviral therapy. The safety and toxicity profiles of nanoparticle-based antiviral therapies are crucial factors to consider for their translation into clinical practice. While nanoparticles offer great potential, their potential adverse effects must be carefully evaluated. Nanoparticles can interact with various biological components, including proteins, lipids, and nucleic acids, potentially leading to unwanted side effects. Furthermore, the physicochemical properties of nanoparticles, such as size, shape, surface charge, and surface coating, can influence their toxicity and biocompatibility. It is essential to thoroughly assess the biocompatibility of nanoparticles, monitor potential accumulation in vital organs, and understand their long-term effects on the immune system and other physiological processes. Robust preclinical studies and rigorous safety evaluations are necessary to ensure the safe implementation of nanoparticle-based antiviral therapies. By designing nanoparticles with reduced toxicity and proactive consideration of safety profiles, the translation of these therapies from bench to bedside can be expedited, benefiting patients with viral infections worldwide. Nature has inspired the development of bio-inspired nanoparticles that mimic the functions and structures found in biological systems. These nanoparticles can demonstrate enhanced antiviral efficacy by leveraging natural defense mechanisms against viral infections. For example, nanoparticles can be functionalized with viral receptor mimics, preventing viral attachment to host cells and interrupting the infection process. Furthermore, nanomaterials derived from natural sources, such as plant extracts or marine organisms, can exhibit intrinsic antiviral properties that can be harnessed for therapeutic purposes. The use of bio-inspired nanoparticles presents a novel approach to antiviral therapies, harnessing the innate wisdom of nature to combat viral infections. This emerging field holds promise for the development of effective and sustainable antiviral treatments. RNA interference, RNAi, represents a powerful mechanism for inhibiting viral gene expression and replication. Nanoparticles can be utilized as carriers for small interfering RNA, siRNA, enabling targeted delivery to infected cells and efficient gene silencing. The incorporation of siRNA within nanoparticles protects it from degradation, enables sustained release, and improves cellular uptake. By specifically targeting viral RNA sequences or essential viral proteins, siRNA-loaded nanoparticles can effectively inhibit viral replication and reduce viral pathogenicity. Moreover, the combination of RNAi and other antiviral strategies, such as targeted drug delivery or immunomodulation, holds immense potential for synergistic antiviral effects. The rational design of nanoparticles for efficient siRNA delivery, stability, and intracellular release is an active area of research. Despite the tremendous potential of nanoparticle-based antiviral therapies, several challenges hinder their clinical translation. One major challenge is the lack of standardized protocols and regulatory guidelines for nanoparticle synthesis, characterization, and safety assessment. Additional challenges include scalability and reproducibility of nanoparticle production, as well as cost-effectiveness, which are essential for large-scale manufacturing and commercial viability. Regulatory hurdles and concerns regarding long-term safety, toxicity, and potential immunogenicity further impact the clinical translation of nanoparticle-based therapies. Collaboration between researchers, clinicians, regulatory agencies, and the pharmaceutical industry is critical to address these challenges. Continued efforts in standardization, optimization, and risk assessment will pave the way for the clinical implementation of nanoparticle-based antiviral therapies. The future of nanoparticle-based antiviral therapies holds immense promise, with several emerging technologies on the horizon. These technologies aim to further enhance the specificity, efficiency, and safety of nanoparticle-based targeted delivery systems. Emerging technologies include the development of multifunctional nanoparticles capable of simultaneous drug delivery, imaging, and immunomodulation. Moreover, advances in nanomaterials, such as quantum dots and metal organic frameworks, offer unique properties that can be harnessed for improved antiviral efficacy. Furthermore, the integration of nanotechnology with other fields, such as artificial intelligence and microfluidics, holds exciting prospects for personalized antiviral therapies and point-of-care diagnostics. As these technologies and approaches continue to evolve, the field of medical nanotechnology will contribute significantly to the development of innovative antiviral therapies, combating current and future viral threats. In the conclusion section of the video on nanoparticles for treating viral infections, it is evident that medical nanotechnology holds significant potential for targeted antiviral therapies. The discussed topics have elucidated the mechanisms, strategies, challenges, and future prospects associated with nanoparticle-based antiviral delivery systems. This exploration has delved into inhibiting viral replication, enhancing drug delivery, surface modification of nanoparticles, and utilizing bio-inspired designs. Although clinical translation and safety considerations pose challenges, the future of nanoparticle-based antiviral therapies appears promising. Through ongoing research, collaboration, and technological advancements, personalized and effective treatments for viral infections may soon be attainable, benefiting individuals globally.